Ja, jag tror inte det.
Anybody else who's left? Is everybody back? A couple people left. Oh, here they go. Okay, one student didn't manage to present before now, so here we have Prakash.
Yeah. So let's start.
Hang on a second. Let's see. Yep.
Hi everybody, I'm Prakash from Nepal. You just imagine from where I am. I'm from Nepal. It bears all the climate types. It starts from tropical to alpine. The altitude ranges from 60 meters above sea level to 8,848 meters above sea level. It's a recent biodiversity because all the climates we can find over there and habitat of many endangered flora and fauna. I studied previously botany and I had a lot of experiences, field experiences in plant ecology. Yes, these are some of the research grants that I found to do in my research, and these are some scientific publications that I have done so far. I spent my childhood in dense forests, beautiful rivers and streams, and my country is rich in biodiversity. I'm always curious to know about the fact about, or I'm always curious to answer the questions of ecosystem functioning, and I'm regularly involved in the fieldwork as a biologist, and I like to learn more about the principal and practical field of plant ecology and environment, so I choose biology. That's all, thank you.
Okay. That was 1 minute 8. Okay. So there's a couple of teaching assistants who are also going to be presenting because they've been either on field work or doing something wonderful. Now I'm just going to go through the stuff here. On Friday, you will be made into groups and you'll start planning, okay? So, and we're still making the groups, almost following the principle we had before. And now there's some changes to that principle, and we'll see how that goes. But on Friday, you'll be finding out who's in which group. And basically, for those who are going to be teaching, we've got this principle that we're trying to apply. We do our teaching like we do our science, okay? And this is often a very remarkable thing for people to think about. We do our teaching like we do our science. That means you guys are actually part of the experiment, which is probably not the best thing to do to tell you this, in terms of psychology not, but it means that we are able to scientifically test out what works and what doesn't by the way we structure this class, and also by the way that we get the feedback here. So some of the advances that are going to be made in teaching methods are coming from applying scientific principles to the structure of this class, and that helps to bring us forward. There's a lot of stuff that you bring to us as well, and on Friday you'll get one of the gifts from a class 2 years ago. But there's a lot to be said here. Now, I'm going to be going through how to prepare for this, because each one of you bears the individual responsibility for carrying this. Regardless of what genius is in your group or how genius you are, you can't slide away. And because your ability to contribute to the teamwork is also a factor on your grade, take personal responsibility starting now. Okay? And frankly, this group has impressed me. There's a bunch of people with lots of really relevant experience, a bunch of people who already have publications or have done the writing and all this stuff. So basically, you have, in fact, the best starting point in years. So let's see what can happen out of this. Field trips cause rain, and this is a Bergen thing. So probably on your field trip, it will rain. One of the things that we're going to be doing now is teaching you, this whole course is making you write a mini master's thesis, basically. That's what it is. But a master's thesis with a purpose, and that is to answer Baden Commune's questions about the water quality. There's a lot of things we could have told you about. Or there's a lot of things we could have taught you. You know, this species here, this species there. But in terms of a general skill, how long will you last without the computer? You'll last a long time. And how long will you last without food? You'll last about a couple weeks maybe, in fact. But you won't last long without water. And one of the reasons I think about this is I was, remember on the first day, somebody said, and she speaks some phrases of Hausa. This is up in northern Nigeria where the Boko Haram are now. And when I was 23, I was visiting a Canadian friend of mine who was a Canadian University Services overseas worker working up in northern Nigeria on the border to Chad. And of course, you know, I thought, well, haven't been there yet, let's go. And so I was up there where there is electricity maybe 2 hours a day, running water, run and get it. You get it from the well, then you filter it, then you boil it, and then you can use it. A market where you had to go and get things fresh every day, and I don't blend in. So I'd have to go to the market, and I learned the numbers first in Hausa, so I could barter. And then because I was coming every week, you know, I'd get what I was paying for, and then I'd get a dash, a little bit more than I was paying for, to come back to exactly that seller. And this was also where it's fairly desert, as you probably see from the news. And there was even the Fulani. You know who the Fulani are? Maybe? No? Anyway, they're still a nomadic tribe that basically deal with cows, with cattle, and they drive them from place to place. And the Fulani men are the one who wear the makeup. And this is really cool, because I'm there in the market, you know, shining like a light bulb, and the Fulani men come in, and they have their hair braided like this. They have wonderful accoutrements and decorations, and they're tall and they're proud, and they know they're good looking. And they're looking at me, looking at them, and they know that I know that they're good looking, and they know that I know that they know that I know. And it was a complete reversal of sex roles and so on. So it was really cool. However, one day, I'm at home, I'm making dinner in the 2 hours of electricity. I'm listening to BBC World Service. And it says, oh, there's been a military coup in Nigeria today. And I'm like, shit, that's here! And a military coup is not to be played with. And basically I stayed indoors for three days because all the village knew I was there, but they didn't want whoever was going to be taking over to know I was there. And then it was a matter of being able to get from the north to the south to Lagos, not my favorite city, and get out on the date that my plane ticket said. And it was, you know, as those 3 days were passing, there was motorcycles with guys on board that had suitcases full of currency. They were heading for the border to change that money for another currency, because the first thing the military coup does is change the currency. And there were armed guards and this and that, and it was a pretty tense situation. And we really didn't know if it was going to be bloody or not. And we also knew that transportation was now pretty hard to come by. So I had to get from the north to the south. I took my remaining money out from under the bookshelf, and we caught a local taxi, which you hail by doing this. That was going to Maiduguri, which has also been in the news, and the airport there. And as we're driving, for example, from Michika, where I was, to Maiduguri, I'm in the local taxi. The local taxi takes everybody and their goats and everything like that. You know, so it's just jam-packed. And I'm sitting in there, you know, like a light bulb. And we get to roadblocks where there's village idiots who have machine guns and basically can't read. And they now have the power. And they stop the car all the time. And They take one look at inside, I'm the person they're looking for, or whatever. You know, I'm the person they're curious about. And the rest of the taxis go, oh God, did we have to let her in? And they say, passport. And I lie. And I say, no, I left it at the embassy in Lagos. The embassy does know I'm there. And so I pull out my student card, which says student in big letters. And I pass it to them. And they look at it, and they flip it over, and this and that. And they can't get me on this. But I know that if they take my passport, I'm screwed. And they let us go. And this happens several times. Anyway, but in the meantime, it takes me a couple of days to get to Lagos. From that experience, I mean, I was really scared. Really scared. And there's a lot of stuff that just is repressed. But after that experience, I know there's 3 things I need. I can walk away from everything else that I have gained in my life. I can walk away from everything. I need clean water, food, and a safe place to sleep. That's it. That's it. So clean water is paramount. Clean water is paramount. When you get down to any kind of apocalyptic this and that, data is not going to save the world. Clean water is. It's that basic. So one of the motivations for getting you this kind of process is basically you will be able to evaluate water quality. You'll know how to test it to a certain degree. And it's some kind of skill that is useful anywhere. Absolutely anywhere. And basically, that's it. That's why you're doing this. Because I know you need 3 things. Clean water, a safe place to sleep, and food. Almost in that order. So anyway, that's the motivation for teaching you this. So it's one of those transferable skills. Great. And the other thing is that how many have written a thesis before of some kind or another? Oh, yeah, okay, let me put it the other way. How many have not written a thesis before? Yeah, that's more hands. Okay. And one of the things that happens is that you get writer's block. And you're not quite sure where to go, and you don't know what the process is, and so on. And since the university reforms here, you have 2 years to go from this end to the other end with a thesis. And without the practice in writing, your chances of hitting the other end without asking for a delay or an extension, and your chances of getting something good on the outside are reduced. For those who have experience writing some kind of thesis or whatever, yay, help your group members. But for those who don't, this is a really valuable lesson. I could stand here and tell you about this all the time. You'd still not learn. It has to be literally embodied. Go from the physical to the mental to the product, and then from the product again to the physical. That's what you're going to have to do. And it's a long process, but it's something you only really understand when you've done it. Okay, and there's this. And this part here, overcome barriers. There will be, but complete the projects. Okay, and this part, it's a cliche, but you're going to experience it. Everybody does, absolutely everybody. But that's why we emphasize teamwork and how to get through the teamwork. Because it's going to be difficult. At some point or another, it's going to be difficult because it always is difficult. Even if you've got the best possible members in your group. So, you will be supported by your PhD guides, your teaching assistants. And this year there'll be 1 per group. Some of the teaching assistants have done this before, and some of them have not. But they're getting guidance also from us. Okay? And so on Friday, you'll be meeting with them, and you're discussing the project design ahead of the field work. And you'll be collecting the field gear, and you'll be doing and planning all this. Okay. So basically, there's field and laboratory work on the field day. On your schedule, you probably saw that there is field work for 4, possibly 5 days. And it'll be the 5 days. And you will get 1 of those days. And those days are efficient because the E. coli samples that you take in the field have to be delivered to Bergen van Lab before 1 o'clock. And that's hard to do. So that means you have to plan with that logistic problem there, because they have to take the samples, plate them out, mark them, and then incubate them overnight so that we get the results back fairly quickly. And it also means so that the bacteria in your samples are not growing while it's cooking in the sun on the field site, okay? So there's your first logistic restriction. You have to get the E. coli samples back to Van Lab. Somebody has to deliver them by 1 o'clock. And they're over in Fyllingsdalen, and we'll show you on the map, whatever. We've also already booked cars from the car sharing ring, because the university has also found out that it's too expensive to keep its own car. We're using the car sharing ring. Everybody know what this is? Oh, okay. Those who know, yeah, go ahead.
Well, it's just anyone who has a driver's license can just rent the cars.
A bit louder.
There's different cars all around in Bergen, and then you can sign up to this website and basically rent a car for a certain amount of time, and then you go and pick it up.
Right. Right. And I'm a personal member of the car sharing ring, and I like to say I don't have one car, I have 32. And these are everything from moving vans down to 2-seaters, and they're parked at different places around the city, but there's a couple of them that we like to use, which is our orientation right now. We are, okay. The Mölndal, just past Café Brenneria over here is a garage, and there's 2 cars parked in there, and we'll be using them, I hope. Yes?
What kind of driving license do you need? Is it a
Valid.
any? You don't need like an international one. If I have a Spanish one, would that work?
Apparently, yes.
Right. Okay.
Yes, yes. So we've booked 2 cars for each of the sampling days, and that means that there may be logistic problems, but you TAs and you work this out, okay? And most of the sites are very close anyway. Not all of them, but most of them. Okay. So that means that it's true, you're only really a half day in the field. So it's early morning, and that day is really well planned. That means you've already got the equipment, you've checked out that the equipment works, and you will be returning it all in good working order. You also have to send or bring the phosphorus samples to Eurofins, it's out in Sandviken, and there is a way that they like to have the standard sampling sheet. I'll show you on Friday, just so I don't get mixed up here. Okay, and of course it's right. So you plan and carry through the writing process, and that's where your TAs are actually helping a lot. Because the first thing you'll be doing is materials and methods, writing that chapter, materials and methods. I'll be guiding you through that. And then in order to, in fact, this is the biggest job of the teaching assistants, correcting the writing. We'll be doing it chapter by chapter. First, materials and methods. And on your schedule, there's a deadline for submission of the first draft of materials and methods. And then the teaching assistants go through it, mark it, I double check them, and then they hand it back to you. You will never in your life get as much feedback on your written material as you do in this course. Never in your life again. But you need this feedback in order to be able to understand what kind of mistakes you make and why they're important, okay? That means that by the next time there's a revision of the materials and methods, and you'll be handing it back to the TAs. And then the results are starting to come in as the E. coli results come in, as the phosphorus results come in, as your biodiversity results have been worked up. And there will be a special lesson on the 31st with Christiana Todt about how to use the biodiversity indices. Everybody really must attend that one, because it's one of the ways in which you can easily figure out if a body of water is polluted, depending on what you got there. And it's not that easy anyway. So Christiana, she's a postdoc or more, a researcher now at the university, and works also with what's called rådgivende biologer, advisory biologists, a company that's been built up by almost nothing to a large and stable one that advises the municipality and anybody else who wants to know anything about the biological aspects of what they're seeing. So these are professional biologists who made a company that now is making money off of advising on biological questions. A lot of it has to do with the water bodies around here. So Christiana is quite well respected and quite well organized. So she's from Austria and really knows her stuff and has a plan for how to do even the lab exercise that you will be getting on the 31st. So, and with the writing process, it'll be chapter by chapter until we get near the end. And if anybody has even looked at the schedule, things are very detailed planned at the beginning. Then we get the field work, and there's still some work to be going on. It's going parallel now. And when we get to the end, you will see that some of the things you've been learning in class, you've also been applying to the reports and the field work that you're working on. We're putting time in class for you to be able to work on the reports. So it's not as though the reports are your homework. The reports are your main focus. And we'll be giving you time to work with that, but we'll also be giving you training in how to write for different audiences, because that's important. Okay? And the oral presentation, there's already been some stuff about that. And it's about 12 minutes. This is basically the entire instruction of what you're going to be doing for the oral presentation. The criteria for all the groups is keep the time limit, get your point across. And that means you've worked through everything about your site. You know the pros and cons of your data. You know what it means. You've decided on what is the point. And that's hard to do. That's very hard to do. What is the point? What do you want to get across? And then you get it across. That's basically it. 12 minutes. That's a conference. That's a regular conference slot. Get your point across. There's many who don't do that at a conference. And you want to get the point across in a very understandable manner. And we'll absolutely get back to that. So, there we go. So this is the background research. When you get there, when you get a site, there's previous information and so on. We'll be suggesting some websites, and there'll be study info and maps about your site that will come partly from, if it's been a study that's previously done, it'll come from that. If it's not, it'll come from your Bergen municipality guide, who will be with you out in the field, okay? So that you know that you're going to the right place, and so that questions you have when you get there face-to-face with the study site, that they are answered as best they can, and you get a context. So you get not just the assignment, so to speak, you get what they're thinking around it. One of the things is this, because you're only going to get 10 E. coli samples. That's all that can be equitably afforded, unless we decide that there's a reason to get more. 10. And so that's why your location is important. Your location, your study sites within or along the water body is important. So you have to think about that. Okay, and when you get the groups, this is on Friday now. When you get the groups, you're going to have to study what it is you want for the focus, and what are the limiting factors. Among them, time, obviously. Among them, time is a limiting factor for you. So the project design. These are your basic questions. On Friday, you're going to have to begin to answer these questions. Design how it is you want to approach it. When you get to the site, what do you do? What samples, and so on. And set up a preliminary sampling plan. Work partitioning. You can't all do everything. You can't all do everything, just because you've only got a morning. And that means you're going to have to divide among you. Maybe somebody takes the biodiversity samples, somebody takes the water samples, somebody takes the E. coli samples. They're done more or less in the same way. But you have to decide where, And you have to be extremely careful about the labeling. Because if you're scrawling like this, you know, sample 1, sample 2, whatever, when it gets to Vanla, they have only that in front of them. And then they have this, and then they produce a standardized data sheet that gets sent back to me. And I have no idea, sample 1, which group was this? So it has to be labeled with the site name, the date, and then the sample site. I mean, is it sample site 1? Is it the only sample? Is it replicate A? Is it replicate B? That kind of thing. So be really careful and write clearly, because now this stuff comes back from our so-called subcontractors, and they do as best they can based on the work that you did. Okay? And there has been times when things have gotten mixed up, delayed, whatever, and the delay will affect the, because water isn't just water, it's the animals and the chemistry inside, and it'll affect how long those have had to change and to deteriorate. It'll affect the pH, all kinds of stuff. So make sure about that. Check out the field gear. Now, this year, we have Jonathan Soule, and he's in charge of basically all the labs and all the lab equipment. And thank God he's also very organized, because the reason, one of the reasons why we had to put this list up here was because every time we came back after summer vacation, stuff had wandered off, and we had no idea where it went. Now Jonathan takes a very firm attitude to what's supposed to be in the Bio 300 blocker and what's supposed to come back in good shape and cleaned and all that stuff. And it is, at the moment, cleaned and in good shape and all there. Should be. And that means that you should go at least once with your TA and find the locker, coordinate with Jonathan, and make sure that you have at least this. Okay? Because this is your basic. You're going to need transport. You're going to have the cooler box, the sterile flasks, 10 vials for the phosphate samples, a couple of buckets, a pH kit and a temperature sensor. I think we've also got nitrate now, and there should be enough reagent strips to work with. And we've also got pH paper in case the pH meter doesn't work. We have water samplers, which is an extension rod with a clamp on the end, because you can't generally reach out there. And we have a couple of ways of making a randomized grid on the shoreline for looking at biodiversity samples in a randomized fashion around the water body. So there's that, and then there's the waders or hip boots, because a lot of water bodies are shallow, but you just need to get out there. And in some cases, in Stora Lungagården, do we have a boat?
Not that we know.
Not that we know. But that means you have to be ingenious if you're going to get to the middle of Stora Lungagården. Either borrow a kayak or rent a kayak from the swimming pool area there, or borrow one or something. Okay. And in the case of Nordåsvannet, and that's where you mentioned Vågesvai and those kind of things, we don't have a boat there either. But if you can get one, and sometimes we do, then that's fine. And that changes your sampling design, changes your sampling ability. But it also means that logistically that day, you've got a little bit more to take care of. So think about this. And then the first aid kit, you absolutely have to bring one, but usually it's not necessary. Oops, wrong one. There. But you still have to dress appropriately, because you're out there for a long time. And there's probably no bathroom, and there's all kinds of things like that. So, like they say on the Alvin that goes, there's a deep sea submarine called the Alvin that's goes out of Woods Hole Oceanographic Institute in Massachusetts, and it's been used. It was the submarine that discovered the hydrothermal vents, you know, the deep sea vents with all the big, that's the Alvin. And my sambo had also gone down into it. It's like this, basically, and it's cramped, and there's room for maybe 2 people, and you've got like all the gear and the thick walls and this and that. And every sub has call letters on the top, and the call letters on the top of this one are also relevant for your field work. P, before you go, okay? So make sure that you're all right, because you're not going to be close to bathrooms there. And then in the afternoon, you're going to have to do some lab work, and that's the biodiversity. So on site, take photos. Take more photos than you thought were absolutely necessary. Some of them will go into your presentation. Some of them, if you take films, that's fine too. And if you have an underwater or a GoPro, wonderful. Sometimes, if it makes a point that you find out is important, or in fact, if you want to go back and use an underwater camera to make a point that you found out is important, do so. Pictures really, really talk, and they summarize a great deal. So, and you can make a picture into one of those data graphics and so on. So please, please don't spare the digitalization here. And it really does make a difference, because when you're talking on dry paper and making a sort of dry science report, because your report will be in the standard science format, sometimes the picture makes the point for you. And we understand what you mean by 2 small words, you know, that's all. But in the presentation, sometimes it's really, really useful. So these things are things you must note down while you're there, because it gives a context to what it is. It also lets other people who've never been there understand what is the connection between the surrounding water, the surrounding land, the residences, the houses, the industry, and it's all different. It's all different. And then the samples and the interpretation that you get. Okay. So this kind of information, absolutely necessary for your report, and it makes a context for your presentations. Okay. Because these presentations will be done to each other. I mean, you're all doing the same thing: biodiversity, pH, and abiotic stuff. Phosphate, among other things. You're all doing that. But the interpretation of this will be completely different for each group. And so in order to make sure that the other groups understand your context, this information is absolutely necessary. Okay? And you'll find out whether or not, for example, the amount of water flowing into a treatment plant is relevant, because you don't have a treatment plant, for example. Or the time of day, that may or may not make a difference. And the rainfall, that's kind of important. You know, if there's schools, if there's construction, there's a couple of sites that you will find out has major construction on it. And it's affecting how you can interpret, and also the biodiversity. So on the field day, there's the water samples for the E. coli, the coliform bacterial counts. I said 10, it's maximum 20 per group.
21 actually.
21, okay, good. So that means you can get triplicates of 7 sites. And the sterile bottles, there has to be these particular sterile bottles. We've got a couple of hundred of them now. Yep. And they have to be kept cold, so you have, of course, a cooler box. Remember to keep to have the cooler elements frozen before you get to the field. Okay, and proper labeling there. There are, just so you know, we have, and there's every year there's a confusion between TCB and CFU. Okay, and okay, what's CFU is?
In colony forming units.
Right. Okay. Do you want to explain? Okay, I'll just draw it. Okay. So this is the petri dish that has the stroked out bacteria on it, and it's usually, what is it, like so, and then like so, and They streak it across like so, okay? And then they incubate it. And the colony forming units,
It's kind of a count of the bacteria because you can see each bacteria, but each bacteria forms a colony, which you can count.
right. And so when he was saying the EU standards, less than 100 is good, you get that. Okay, and then less than 100, this is what, 10 maybe, 9. This is obviously good. But when you get a lot of them, in fact, this whole thing can be grown over. Colony forming units, and then it's 1,000, okay? Which means it's really hard to work with statistically. And that's why we ask for a dilution. In the case where we suspect that it's going to be higher than 1,000, which is unacceptably high, we ask for a dilution at the lab itself. And it has to be noted, and we have to warn them so that we maybe dilute by 1/2 or dilute by 1/10. And we find this out, and but the reports come back to you in terms of colony forming units per 100 ml sample, okay? So your reports come back to you in a standardized form, and then you'll find out from these columns there whether or not the replicates actually are replicates and whether the dilution made a big difference to the interpretation. So that's colony forming units. What's TCB? You're probably right.
Total coliform bacteria?
No, thermotolerant colony forming bacteria. So thermotolerant, that's that part there. And like so. And this is a bigger concept. So that can be more than just E. coli. And E. coli is what we're supposed to be measuring, and we hope we actually are. But it can be slightly more than that that's in the tray. Okay. But the water quality standards are based on colony forming units, okay? So colony forming units, and that means that is the unit on your axis of the figures that you're making out of this too, okay? Just checking the time. Okay. This is a proper label, for example. So you have UIB, so they can figure out who they have to bill or send it to, which group you are, the site name, which of your sampling sites it is, which replicate it is, and the date, okay? And using a waterproof marker, for those who don't have a waterproof marker, you know, pencils work. Pencils don't wash away in rain and so on, and it'll survive a lot longer than ink will. So think about that, because it's going to get messy out there, messy and wet. On the phosphorus levels, the phosphate, they'll indicate eutrophication, too much algae or whatever, or the possibility of too much algae. You get 10 samples, and they're in non-sterile flasks that I think we have enough of right now. And again, they had to be kept cold, and they'll be done by Eurofins, and the results sent via email. Again, a standardized results sheet that comes back. And if you've got replicates, you've got replicates. They're just processing the samples. That's what they do. And they send them back with a label, with the labeling system that is yours. So if it's not clear, it's not clear. And you have to have the group number and the site name on there, or I won't be able to send it back to the right people. And then you have a couple of things for the field day. There's a couple of pH meters and so on, and Jonathan has made sure that they are in perfect working order. So that's one of the things, and it's supposed to come back in perfect working order. But in the field, lots of things go wrong. It's literally Murphy's Law. If it can go wrong, it will go wrong. And you'll have to tackle it on the spot, and you have no time. This is why you prepare the day before, you double check your equipment. This is why also it's not consecutive days of field work. It's not like group 1 goes out and then the next day group 2 goes out. There's time to deal with any kinds of problems with the equipment. So you have the high temperatures and the differences in pH as compared to the surrounding water. It might tell you something about the pollution source, if that's what it is. And it'll all be done on site. So this is part of your work division. And the biodiversity. Okay. The biodiversity tells a lot. And even though you're going there for a snapshot of the system there, it's just a snapshot. And the chemistry and the temperature and all that can change overnight. But the biodiversity, it lives there. It lives there, and it tells you an awful lot. There are things like indicator species, and you're going to find out whether or not you can find them and what they are. And then you're going to be able to make them up into biodiversity indices, which is what Christiana is extremely good at. You have different ways of collecting that biodiversity, with a grid, with a shuttle, with kick sampling, with all kinds of things. And kick sampling is remarkably common and remarkably hard to standardize. So you do what you do, you write it down, and try and come up with something that is good enough. And you will also find in the literature, kick sampling is what's often mentioned, and again, they've probably done it in a different way than you're planning to do it, or can do it, okay? You'll also come up with some problems like you got a rock, and that's the only thing that's there. Or when you've tossed the grid, and say if you want a randomized section of the shoreline, maybe you have stood with your back towards the shoreline and tossed something, and wherever that something lands is where you're going to place the grid, and then you're going to take a certain amount of the grids in there as the sediment and sort the sediment. That's up to you guys. But the randomization of this is also part of the process. You can describe what's around there for the biodiversity, but in the soil, it's different. And then if you want to do the plankton sampling, you can do all kinds of things. And you can do grab sampling for some marine sites. But don't underestimate this. This is a lot of work. So there's some different kinds of sampling that you all get to. Who's been using these sort of grabs? Anybody? Yeah. What? Okay. Yeah. I'd like the two of you. Which grabs did you use, and which ones did you use?
Van Veen.
You used the Van Veen. What were you doing with it?
Ventus something.
Yeah. Okay. And it was just basically it's.
It was like
Yeah, it's this guy here. It comes down and then it hits and then it locks and brings up things like that. Of course, if it locks on a stone, it doesn't close properly and you have to do it again. How deep were you going?
I think about 200 meters.
Oh, wow. A lot of, if you made a mistake, it's a long way back
Yeah.
up. Yeah, yeah. And you?
Half an hour.
Yeah, I know, yeah. And you?
Just the Niski bottom.
This guy? Yeah.
So we measured water in different tidals. So every hour we went out and measured what was in the water. We used to get just above the ground, so nothing disturbs the ground.
Right.
And we took everything from the
Okay.
temperature and biodiversity out of it.
Right. Yeah. Really useful things, those Niskin water samplers. Really useful. Because you can close them at exactly the right depth you want as well. Expensive, but really useful. And if we can, we also, Magnus, do you want to talk about the ROV? I don't know if I've got it here. No?
I don't want to talk about it before it's ready.
Okay. Yeah. And then the field work. This is for the lab work, and you'll be getting a brief introduction to this on the 31st. And the lab work, you'll be surprised how long it takes and how difficult it is, because basically you're going to be counting the number of individuals per species and group. So think about this before you've actually gathered 3 buckets full of stuff, and you're going to have to sort through it grain by grain. Okay, so think about your sampling design. Think about what you want how big a chance you have of actually getting through your sample. And it'll look like this to begin with, and then it gets sorted out into the petri dishes, and then you start counting and identifying. And that ends up being a lot of work fairly quickly. If you're doing this zooplankton counts, you'll end up with, for example, a number of species per volume, because you'll know how much water you took, if that's where you want to go. And the counting chamber. This is just so that you can have a drop of water that gets, or a little volume of water that gets spread around here. And even though, for example, the animals might still be alive and could swim from place to place, you can follow under the microscope that you've counted that, you've looked at that, and so on, and you can follow that you've done the sample without having to repeat some areas and forget some others. Okay, and the bacterial counts. This is just to show that these are coliform, which is coliform, which is the same thing. And then there's something different in this case. It colored differently because of the reagent. The E. coli was there. So just so you know that there is a difference. But we're looking at the bacteria in general, and we're making very often the synonymization between colony-forming bacteria and E. coli. And there's an EU water directive that you'll be able to look up and find. There's also a Norwegian water directive, Water Quality Association, which is in fact stricter than the EU one. So whichever one you refer to, it has to be stated. And you will, of course, be able to compare between samples and guidelines and all that stuff. And you'll also be able to test if the differences are significant or statistically relevant, which is why we don't want more than 1,000, because you can't test that. We want the dilution and the numbers that you can test. Okay? And for the data analysis with the phosphate levels, you'll be comparing the phosphate levels between sites and with results from the previous years. And this is now where the literature starts to come in, because there's no easy table like there's for E. coli. But you'll be able to look at some other water bodies where the phosphorus has been done, and whether it's comparable to you, and basically whether or not yours are higher or lower than is normal. And for the group that gets lille lungegårdsvann, think about this. And then you have these ones, which can be compared and tested for significant differences, and either in depth or across space or across time, and the biodiversity. So you'll be comparing the species richness and the total number of individuals between your sites and then for the previous years or other places. And you can test for differences. So, last little bit here. You'll be getting much deeper information on this from Christiana on the 31st about the indices that you get. You'll be using, for example, an ASPT, the average score per taxon, which is based on other literature and a lot of work by a lot of biologists, or a biodiversity monitoring work package score, which is also something that's been internationally worked up, but hasn't been worked up for all species. It's coming, though. So there's also a Shannon Biodiversity Index, which you'll be working up. And it tells you the relative abundance and the taxa and the evenness. Is it like just one nematode right there and that's it? Or is there a bunch of nematodes and a bunch of clams and a bunch of, worms and all that? So it tells you the evenness. And that tells you something more about the way that life can go on at your site. And basically the diverse, they have a higher Shannon Biodiversity Index than the taxon poor. But basically, in Norway, it's taxon poor. It is in no way as rich as some of the countries you've been in, and particularly the Southeast Asian ones, where every time you turn around, there's a new plant or a new animal, or So Norway is really simple this way. There won't be that much biodiversity. But this also means that the biodiversity that you encounter out in the field or wherever you go to will be able to tell you again something more about the viability of the site you're in, wherever you are, wherever you are. Okay? And then you're going to have to make the interpretation and the discussion. And this means that you start discussing right away. Right away, as you're out there in the field. And you've made notes, God help you if you haven't made notes, because you're going to forget. And you make notes about every little thing you do. Much better to have more notes than too few. And 2 months from now I say, but did you do this or this with your pH meter? Did you hit the bottom or what? Things like that will make a difference. And you can't remember, or you think you did, make a note. Or for example, especially with the E. coli samples, you're out there with your rod and you dropped it and you've inverted the flask and so on, but you were really close to the bottom, and maybe there's still suspended stuff down there, and that's going to influence your results. So make notes, and this is one of the reasons why we want you to discuss your field work beforehand, because the question for each site is clear, and I'm going to load up each site's description to the website right after this class, about now. And how can you explain your results? Include your observations, so on. Use diverse literature and your brains. Okay, you were supposed to be the smart one in the family. Show me. Okay. And the other thing, this thing about use your brains, it's very, very useful. Right now I had one and now a sequential one out at a particular farm.